Hey everybody, hope everybody is doing well. Here we are in our second week of remote learning. Okay, so if you need to, I would review the last video I put up on RNA and transcription. So for a recap, DNA is a double-stranded polymer that's found in the nucleus that has on it the coded information for the proteins that are made in a cell. A gene is a length of DNA, or a length of these nucleotides, that actually has the code for the protein to be made. If you remember, proteins are made of amino acids. So the code on the DNA is going to determine the amino acids that are found in my protein. And therefore, what a gene says, or the protein that will be made, is going to depend on how long the gene is, so that's the length, and the sequence of the nucleotides, or the order of the nucleotides. Now, we also need to keep in mind that DNA cannot come out of the nucleus. So therefore, the information from the DNA will be transcribed onto a strand of RNA. What that means, remember, is that a strand of RNA will be made from the gene on the DNA. The base pairing rules between DNA and RNA, you still keep your C's with your G's, but an A on DNA will pair with a U on RNA, and the T on DNA will pair with the A on RNA. Okay, keep in mind that you can go back and replay any of these videos anytime that you need. Okay, so there is this hypothesis called the one gene, one polypeptide hypothesis. Another way to say that is one gene, one protein. What this simply means is that every gene on the DNA in your nucleus codes for one protein. Reviewing DNA that's what your chromosomes are made up of. Okay, so let's get down to the facts here, or the basics. On DNA, we have a long strand of nucleotides, and this long strands of nucleotides makes up the gene. But it is a sequence of 3 nucleotides that code for one amino acid. Now, since there is only one difference in each nucleotide, I can say either each 3 bases of DNA will code for one amino acid, or I can say a sequence of 3 nucleotides of DNA will code for one amino acid. Keeping in mind the only difference in one nucleotide compared to the next is the base is different. It's either an A, a T, a C, or a G. All right, so genes code for the type and order. AA here is amino acid. So the gene is going to code for the type. Which amino acids do I have? And the sequence or the order that they appear. So, using the information that was transcribed onto the RNA, or remember, the RNA that was made from the DNA, out at the ribosome, we are going to use that to make or assemble, that's what assemble means, a strand of amino acids to make a protein. So the process by which the information coded on the RNA is used to assemble amino acids in a particular order is translation. Translation is converting the code on the RNA to a sequence of amino acids in my protein. We are going to really focus on messenger RNA and transfer RNA to form the polypeptides. And remember, polypeptides, that's a fancy word for proteins, as directed by the coded instructions on the DNA. Wow, that was a lot, huh? So, really simply, the DNA is in the nucleus. That information will be transcribed onto a strand of messenger RNA. The messenger RNA is single-stranded, remember, and it can come out of the nucleus. It goes to the ribosomes. At the ribosomes, I'm going to translate that code, and I'm going to dictate from that code the sequence of amino acids in my protein. Now, I need the amino acids to get to the ribosome. Here's where the job of the transfer RNA is going to come into play. Transfer RNA carries amino acids. The amino acids are present in the cytoplasm. They are there from the digestion of proteins and the diffusion of amino acids into the cell cytoplasm. Okay. All right, so we have a couple of words here. Codon. A codon is the base sequence on the messenger RNA. And the codon codes for one amino acid. So you take 3 nucleotides on RNA, and they have the code for a single amino acid. Since the only difference between one nucleotide and the next is the base, I can say each 3 base sequence on messenger RNA codes for a single amino acid. So, how do I know which amino acid is coded for? Kind of lost my words there in a second. So here's where we have, and this is my favorite part here, I wish I was teaching this live, the secret decoder ring. So this is a ring, and on the outside of my ring, let me get my highlighter out here, these are all the names of amino acids. You are not responsible to remember those names. They will always be given to you in one way, shape, or form. So I'm going to start with a particular code that I find on my messenger RNA at the ribosome. You start at the center ring. So I'm going to start, let's say I start with the G, and I work myself out to, so my sequence of 3 is G, C, U. Well, that codes for the amino acid alanine. You don't have to memorize this. This would have to be provided for you. Second, and show you the start codon. All right, so start codon, and let's use a green highlighter. A, U, G is a start codon. Every single protein will begin with AUG on the messenger RNA. So let's look at the making of a protein at the ribosome. I'm always going to have a start codon. Let's go back here. All right, so let's look at the ribosome as a scanner. My messenger RNA will get placed on the ribosome, and the scanner will go until it reaches AUG. AUG actually codes for an amino acid, methionine. It's always the first amino acid in every protein. And then I'm going to scan, and I'm going to read off. It's like a ticker tape. I'm going to read off every 3 bases and assemble my protein on the ribosome until I hit a stop. So every single strand of messenger RNA has a start and has a stop. All right, so let's take a look at this. I will put a video clip of this for you also. So I'm going to start up here. Let me see, red highlighter, I guess. It's the only color that's not here. All right, so here's my AUG. That is my start. My transfer RNA carries an amino acid called methionine. All right, so then I'm going to scan down to the next set of 3. My next set of 3 is GCC. That is the code for alanine. You don't have to memorize that. I would have to give you a secret decoder ring to do that. And then a bond will form between methionine and alanine. My next transfer RNA would come in, and that is going to be the codon here is UGG. Transfer RNA comes in, and it's tryptophan, and I will form a bond. This is the beginning of my synthesis of my amino acid sequence. Okay, so showing you another way. Looking at this picture, I'm always going to start here at the AUG and bring that amino acid in. And then I'm going to just follow along, and I'm going to match up letter for letter, following the base pairing rules. And these dots here on my transfer RNA, these are representing my amino acids. And this will go on until I get to the end of my messenger RNA strand. So, the transfer RNA is in the cytoplasm. It has the code for a particular amino acid on it. It will only, UUU will only carry lysine. It will not carry anything else. And we will follow base pairing rules, and that will go in right here with AAA, and that will place lysine next in my sequence. We call this UUU here the anticodon. I'm just mentioning it here. I will never ask you to use that for anything. All right, so in this diagram here, this blue blob and this gold blob represent the ribosome. This is my messenger RNA. This is my transfer RNA. This is my amino acid. And notice my base pairing rules. A with U, and U with A, and G with C, and U with A, and U with A, and C with G, and A and A and A with U and U and U. And then so on, until I assemble my protein, and this will go on until I hit a stop. All right, so this is a lot, I know. You can watch this video as much as you want. I will be a video chat on Tuesday the 24th at 1 o'clock. You are not obligated to participate. But if you want to, you can feel free. So I will put up some review work with this and find a YouTube video that actually does a good job of showing this to you. I hope everybody is well, and stay in touch.